it is my honor to invite Dr. Bijay Patni sir, a respected cardiologist from Kolkata with over 35 years of clinical experience. Sir will present his talk on drug microbiome synergy, how tizapidide reshape the gut ecosystem. Please welcome Dr. Bijay Patni sir.
Thank you, sir. And first, at the outset, I would like to thank the team CardApCon, and particularly Dr. Ashutosh Mishra and Dr. Pallavi Mishra for inviting me and giving me a very interesting talk, I would say, because if you go through the literature and whatever we have come across this particular molecule of twincretin, uh, you will realize that we have been concentrating on the receptor part only. Now, what we need to know is how come this particular molecule having this has unlocked unprecedented clinical outcomes, a pleiotropic effect on most of the organs. There must have been some kind of other mechanism which probably is forcing this molecule to have such impact. So that is what we are going to look into and try to look into the gut microbiome and its relation with tirzepatide. How does it really reshape the whole gut ecosystem to get all the benefits? That is the talk of the day. And the talk given to me with the title Reshaping the Gut Ecosystem for Metabolic Benefit with tirzepatide. So this is what we are going to look into. So first and foremost, where do we stand? We have two important molecules. One is the GLP-1 and one is the dual agonism with GLP-1 and GIP. If you try to look into the various trials which has gone, whether it is SURPASS or SURMOUNT, what we have recognized that be it HbA1c reduction or be it weight loss, both of them, tirzepatide scores. What could be the reason behind it? Is it only the GIP component which is giving all the benefits? Probably not. So we need to understand the complete story, the true blueprint behind this dual agonism getting maximum benefit, the unprecedented outcome which this particular molecule is showing. So here we try to delve into its bidirectional, the outcomes as far as the tirzepatide is concerned and the ecosystem of the gut is concerned. So we'll try to address few issues accordingly. Now let's look at what our gut says. Now we have this slide shows two components. One is the healthy gut and one is the dysbiotic gut, which we find in diabetes or obese patients. Now in a healthy gut, you have an intact mucin layer and a tight junctions. So probably whatever we have, those harmful bacteria is unable to pass through these tight junctions to enter into the bloodstream. But what happens in a diabetic population or in an obese patient, there is a breakage in the mucin layer leading to called as leaky gut. Now that will go through the tight junctions, they become leaky, and as a result, the toxin, that is lipoprotein saccharide, that will go through through these leaky tight these junctions, and then will bind with the TLR4, and that will activate the NF kappa beta and ultimately lead to systemic inflammation leading to endotoxemia because they come into the blood and they can lead to endotoxemia. So this biases will trigger a leaky gut phenotype, elevating the zonulin which breaks the mucin layer and also helps in getting into this episode of breakage of the mucin layer and the tight junctions, and ultimately driving the LPS translocation, which will instigate the systemic insulin resistance and steatosis because of increased inflammation. So this is what happens with the gut when it is healthy and when it is dysbiosis. Now, if you try to look into the various mouse models which tirzepatide used in the phase 1 trial and phase 2 trial, where they try to look into preclinical evidence. What they wanted to show that if you are giving tirzepatide treatment to a mouse model, probably if you look at diabetic model, it is straight, there is no diversity, and this alpha diversity, because of the increase in diversity, probably the gut ecosystem is maintained. So one need to understand with the treatment of tirzepatide, you have more trend towards that going into the healthy control based ecosystem, the gut ecosystem. So that is the diversity part where you have an increase in the gut diversity or the gut microbiome diversity, leading to a healthy structure. And similarly, you can see there is a definite shift in the microbiome shift from the dysbiotic baseline to the normoglycemic control clusters in a mouse model. This is an animal model study, and you can see this was published way back in 2013, which clearly showed that tirzepatide restores the gut homeostasis with microbial shifts mechanistically linked to normalized glucose area under the curve and reduced HOMA-IR, that means it reduces the insulin resistance. So this is how the mouse models showed with the impact of this particular molecule, tirzepatide, that it clearly showed that it maintains the gut homeostasis. Now, when you try to further look into it, what it does and how it does actually, then actually the molecule selectively expands the short chain fatty acids, produces and suppresses the pathobiotes. So basically all the beneficial microbiome is get up titrated, be it the upregulation of this beneficial taxonomy of the various microbiome, whether it is Akkermansia, whether it is Clostridium sensu stricto, or it is Rhomboidsia, all these get upregulated. So these are all beneficial taxa which is there in the microbiome in the gut. And there is, along with that, there is a down regulation of those microbiome which are harmful for the gut, and there is a definite down regulation of bacteroids and the associated other harmful microbiome. So beneficial taxa expansion degrades the outer insulin to expose the fresher substrate and upregulates the GLP-1 receptor. That I'm going to go through it. Basically what happens is that whenever you have this beneficial taxa upgradation or upregulation, you will definitely have the better or towards a healthy gut. And this harmful bacteria or harmful microbiome, they will definitely have an impact and thereby lead to the leaky gut syndrome, increase in the endotoxemia. So now what happens with tirzepatide? Now you are giving external or exogenous tirzepatide injections. So what they are doing is they are upregulating the beneficial bacteria, and these beneficial bacteria are actually increasing the short chain fatty acids, acetate, propionate, and butyrate, which ultimately on the intestinal L cells binds to the FFIR2 and FFIR3 receptors, which ultimately will lead to the increased endogenous satiety hormones along with GLP-1, PY release. So ultimately what you see is that this is basically a vicious cycle. It's no more a bystander, what I would say, this particular microbiome. They are active participant, because what they are doing is whatever exogenous tirzepatide administration we are giving, they are actually this increase in the short chain fatty acid and increase in the beneficial bacteria ultimately lead to more increase in the endogenous GLP-1 receptor, the GLP-1 release. So ultimately it augments the whole phenomenon once again. So expanded microbial communities raise the luminal short chain fatty acid concentration, extending the pharmacological efficacy, what we know beyond the dual agonism. So now we are very clear about it, what could be the reason behind the ever expanding benefits what we get with the dual agonism of this particular molecule terzidopides. Not only the receptor agonism, but along with that its impact on the gut microbiome. Now, along with that, obviously, we aware of that there is definitely a multi-organ metabolic reset which happens along with this particular molecule, and that is coordinated with the bile acid chemistry and tightened intestinal junctions. These are the two mechanisms where you can maintain the integrity of the gut, and you can see whenever there is a primary bile acid that is changed in secondary bile acid, which actually lead to the activation of the FCR and which will suppress the de novo lipogenesis, and then again there will be a TGR5 activation. So this will definitely increase another, this is another way of increasing the GLP-1 receptor release, GLP-1 release. So basically what we see in one side that the bile acid biotransformation has an impact on increased GLP-1. Along with that, if you see the barrier fortification, again the similar step which I've already talked about, there is a decreased serum zonulin, which actually maintains the mucus layer. As a result, because of the increased mucus production, the junctions are not harmed. So basically, there will be halted LPS translocation. So the endotoxemia is taken care of. So that what happens with microbiota mediated bile acid chemistry. So you have a fortified barrier which cut off the endotoxic drive, creating the favorable conditions for insulin sensitivity and lipid metabolism. So this is the mechanism through which probably terzidopaide act as well. Now, we are all aware of the impact of this GLP-1 molecule, this GLP-1 GIP molecule. So how much of its impact can be on the weight loss and how much of its impact can be of insulin sensitization just because of its bidirectional behavior with the gut microbiota. Now, thus gut remodeling which occurs as a result of this particular molecule, that can be the primary driver of metabolic normalization to the extent that almost 30 to 40% of weight loss which we observe, it because of the integrity of the gut epithelium. And along with that, if you see insulin sensitization increases by 30 to 50%, and that is also contributed by these microbiome pathways. So quite an effective molecule just through this mechanism of gut pathways. This particular molecule can act to this extent, almost around 50% of the weight loss and 50% of the increase in the insulin sensitivity can be contributed to its impact on the gut microbiota. So Robutia stimulates the PYY and CCK for profound satiety. We all know about it. We have showed it on the previous slide only. While Akkermansia-derived propionate inhibits the hepatic glucose production by the AMPK pathway. So both of them are beneficial in having the impact on weight loss as well as the increase in the insulin sensitivity. Along with that, what we have learned from Synergy NASH, that there is hundreds, if you just look at this step ladder chart, you will see that there is a 62% mass resolution rate which we found in Synergy NASH, that there is a hepatoprotection which is amplified because of the decreased portal endotoxemia and modulated lipid metabolism which occurs through the this particular molecule interacting with the gut microbiome. Because if you have a reduced pathogenic translocation of the toxins like LPS, then probably you are going to reduce the inflammation, and thereby it decreases the hepatic inflammation and the fibrogenesis. So obviously now it has a hepatoprotection impact as well just by gut modulation. So this again is a clear-cut way of understanding that this terzidopaide can have an impact on the hepatic protection through the modulation of the gut microbiome. If you look into the kidney issue as well, that is the renal protection impact of the terzidopaide. Now that is also microbiota dependent, which again can be seen through these two particular graphs if you see. Now if you see this, the intact gut, what you see, if you are giving a terzepatide, you will see an improvement both in the EGFR as well as albuminuria. But suppose you are given an antibiotic and that has depleted the gut microbiome, then you will almost have no impact, even if you give terzepatide. So you have a flattened kind of impact both on the EGFR albumin reduction as well. So this is again a pointer to the fact that these gut microbiota is no more a biomarker. It perhaps had an active participant in this particular, when it tries to interact with this particular molecule. So you can very well see that if you have a loss of gut microbiome, the beneficial microbiome, then you won't have much of a renoprotective effect even when you are trying to give terzepatide. So resolving bacteroids dominated dysbiosis reduces uremic toxins, while short chain fatty acid upregulates the anti-aging renal protein. This is extremely important because both the hepatoprotection as well as the renal protection is basically through the various interaction which the terzepatide has with the gut microbiome. Now, another factor is that now this particular molecule, terzepatide, this operates across dynamic bidirectional communication highways. All the organs cross-talk and they have the benefits when terzepatide is given. If you look at the gut-brain axis, you have to look at the gut-liver axis or the gut-kidney axis, the expanding beneficial community produces more short-chain fatty acids and fewer LPS molecules, sending protection signals across integrated gut-brain-liver axis. So when you see this particular molecule, where you have all these particular receptors, GLP receptors and the GIP receptors present in various organs, probably they undergo, they actually interact with each other and then have a beneficial impact on this particular combined axis of gut, brain, and liver, kidney axis. So again, do the various mechanisms which have been outlined here. The acetate increases the vagal hypothalamic GABA signaling, reducing the hunger and drive. The gut liver may decreases the portal LPS, as I said initially, when we are dealing with hepatoprotection, and increase the FEXR activation. Thereby again, it has a hepatoprotective action. And again, if you reduce the uremic toxins, probably you have the renoprotective impact as well. Now, what we talked about various mouse models, now we have certain early human studies as well, the preclinical studies which were undertaken, and these are the two major trials, SURMOUNT-1 and the SURPASS-2 post-hoc, where SURMOUNT-1 was dealing with only the obesity population, where it showed terzepatide actually leads to beta diversity normalization achieved by the week 40 of the treatment. So again, that is definitely the issue of diversity. If you have increased diversity in the microbiome population, then you will have all the benefits. If you look at the SURPASS-2 post-hoc trial, again there is the enrichment of the beneficial taxa, that is the Akkermansia and the Mucinophilia and the Faecobacterium. Along with there is a decrease in the harmful ones, that is Bifidobacterium depletions occurs. All will lead to HbA1c drop along with improvement in the as far as the weight is concerned. So fecal metagenomic sequencing from primary clinical trials like these two has strongly suggested that the gut ecosystem remodeling is a reproductive feature of the human pharmacology. So you can reproducible features. So basically you can reproduce it. We have seen in these two trials, it has been reproduced. So basically again, it clearly confirms that clinical metagenomics confirms the preclinical taxonomic shift and metabolic correlations. Now what the physician need to know, what this can be implemented as far as physician. And that is also essential, but more important is you have to have that proper diet as well, because that is extremely important. So dietary patterns and precision symbiotics can purposefully accelerate the microbiome shift terzepatide induces pharmacologically. So basically, again, this is the process which probably we have to employ in particular individual and approach this as a four-step approach in order to get the best of the benefits. Now, this is the research horizon transitions from correlation to causality. Now, we initially thought it could be a correlation, but now we are aware of the fact that with various trials, whether it's animal model or the preclinical or the early human models, that it is actually causality and taxonomy to metabolics. So basically, you have these four major trials. The synergy NASH I talked about, that phase 3 microbiome endpoints. Now the HMT, that is, fecal FMT, that is the human fecal microbiota transplant, causality studies is going on. Probably it is also being employed. Outside it is employed, have been employed now in India also. We heard that some of the institutes are using it. Multi-omics integration, and then terzepatide with semaglutide, comparative microbiomics. So if you look at this last part, that is the GLP-1 and the dual agonism, whether the GIP is actually principally responsible for whatever change we are getting as far as the gut ecosystem is concerned, the beneficial gut ecosystem is concerned. We have to look into it through various phase 4 pharmacovigilance and FMT studies that will definitely establish that microbiome as an active effector, not merely as a biomarker. Now, you cannot use it as a biomarker. It could be an effective effector, and that is why the bidirectional word comes, that both probably acts and then you get the results. Now, if you looked at the Venn diagram year out, what we know is terzepatide is the center of incretin pharmacology, microbiome remodeling, and end organ protection. We have understood, we have enough of data through SURPASS and SURMOUNT and various other trials that if you want to get the full potential of this paradigm shift which we are seeing, probably you have to understand its deep partnership, the terzepatide's deep partnership with the gut ecosystem, and you have to leverage that. So now we can actually say that terzepatide is a microbially intelligent metabolic therapeutic. So that is what I would like to conclude with, that now we need to look in a fresh light or fresh way of looking into this particular dual agonism, that we are probably missing out on certain important ingredient, particularly the gut microbiota, which is responsible for almost half of the results or the outcome which we get with terzepatide. Thank you, Dr. Asutosh Mishra, sir, for giving this talk to me.
Thank you, sir. Thank you for the excellent talk, and that's what I expected from you. And you delivered right on the target, and I hope this explanation will go into the further understanding of the people who is listening the talk, as well as we will try to propagate this thing via the recorded lecture. The thing is, one very important stratification also comes as a gut microbiota profiling. Suppose we want to address residual cardiac risk. We want to address the residual renal risk. We want to stratify the patient in terms of MSALD. In those subgroup of difficult non-responding patient where their progression is very fast. We can do gut microbiota profiling, look at the alpha diversity, beta diversity, and accordingly we can plan the therapy with the help of FOS, inulin, and other molecules, and we can measure the markers of inflammation and the biomarkers like GDF15. It is very, very important. That is actually a driving factor. You can actually not only stratify these patients into bad patient and good patient, and you can decide upon the therapy, as well as you can switch the therapy from GLP-1 to the twin creatine, as you have already said. So where do you place this molecule? We have lot of evidence in terms of advanced CKD patient where gut microbiota is one of the main steam therapy in refractory cases and in MSLD patients. So particularly these two aspects, the MSLD and advanced CKD.
See, whenever we ask to place these two molecules, the GLP and the dual agonist, you will see most of us, what we understand is that we want to say that most of the comorbid condition, if you have, probably the GLP-1 receptor has more data, right? So now with the newer insight what we are getting, that this has a huge impact on both the hepatorenal profile of the individual. So this should be an important molecule in these group of patients, number one. Number two is that if you can do the initial aspect of looking into the gut microbiome profile, then you can pinpoint or go for the precision kind of medicine and actually shift from a GLP-1 receptor agonist to the dual agonism. And that could be beneficial as well. Third is that what we are expecting from the MMO trial, which is going to come 2027, I think the mortality, morbidity outcome trial with the tazetapide. So that is going to give another aspect where we are going to land up. If that trial is a superior trial, then probably GLP-1 receptor agonism may not be sufficient only. That is important because we are looking into that now. We have got enough data now on the synergy NASH. We have got enough data on the renal protection. We have enough data on the glycemic control, weight loss, obstructive sleep apnea. So all these data are clearly there. Now, if you have this MMO trial, which is coming 2027 out, then probably I would say that maybe we have to switch from single agonism to dual or triple agonism.
Says that future of diabetes treatment lies in the combination therapy of GLP-1. It is not GLP-1 alone. So still this is expected that in the future we may be using more molecules in GLP, GLP-1, glucagon, the FGF23 along with GLP-1, MIU with GLP-1. So this kind of therapy where the GLP-1 combines with the other things may be going to set the benchmark for this. Any other comment or anybody wants to say among you others?
Microbiota transplant, that could be, that study also needs to come out because then we may think again about using any other molecules.
So basically, it is still being studied. Let me tell you, we have not yet got. See, one another important type is type 3C pancreatogenic diabetes, where FMT was tried, still in the phase of getting the right outcome. AIG is doing a big study on that particular part. Now, as far as this particular molecule is concerned, we are still waiting for the results. It is like postulated that this probably could be the way forward. So we need to still wait for the results to come out. that whether FNT can have a right kind of impact as far as the outcome is concerned. But looks like a promising kind of stuff. So I think 2027 you might get the results, and on this particular molecule only, whether FNT after identification of the deficiency in taxonomy, the beneficial taxonomy, if you give a FNT along with terzitabide, you might get the benefits. Might. So still in that stage, I don't know about it. Right?
So, Patni, I don't understand properly. This is a head-to-head trial between FMT and Drug-Microbiome. Yeah, I'll tell you something. This kind of a trial will never see the light of the day because all these are pharma companies and they have their own commercial interests. Suppose FMT turns out to be superior, they stand to lose a lot of cash.
So
After FMT, if this kind of molecule, actually because of everyone in the case of FMT is actually reverting back to the again bad gut microbiota, changing the ubiquitous bacteria ratio. So what they are trying to do is give appropriate amount of correction and then maintaining that gut microbiota with the help of various things like lifestyle modification, the symbiotic, probiotics, and with the help of these molecules. So that is the thing he wants to say actually. For the maintenance and the efficacy of FMT can be increased by this type of molecule.
Yeah.
So these are the thoughts and they are going on.
basically it is a synergism.
Yeah.
It's not that key this versus this. It is the synergism. Once you identify the deficiency or the taxonomy, if you microbiome taxonomy, if you identify, and if he is a right candidate, then probably both of them can help in a better way. That is what they are trying to look into. But let me tell you, very difficult till the actual results are out. Yes, sir, Dr. Akash Singh, what do you want to say? Sir, आप बोले बिना होता थोड़ी है. Right. So, okay, sir.
Thank you, sir. Thank you very much for the very informative and path-changing session. So let's move to the another session.